Hello, my dear doctors, and now we are going to start with one of the most important topics in anatomy, that is embryology. And in embryology, first we are actually going to study about the general embryology part, of course. But in general embryology, first we are actually going to learn about the gametogenesis, guys. And after learning about gametogenesis, that is spermatogenesis and oogenesis, then we'll learn about what's going to happen in the first week and second week and third week of pregnancy. Now, before starting with gametogenesis, that is spermatogenesis and oogenesis, guys, I want you to recall about the cell division, that is mitosis and meiosis first, so that you will get an idea like what are the stages, in which cell division the number of chromosomes are reduced to half, and then keeping that idea in mind, further we can study with confidence the gametogenesis, that is spermatogenesis and oogenesis, guys. Is it okay? I know, like, even though most of the students might be knowing about this part, that is mitosis and meiosis part, but still I don't want to take any risk over here. Let us just try to recall back in our mind. So please write on the heading there as gametogenesis. And in gametogenesis, first let's see what's happening in mitosis. Now, what is mitosis here? In this, what is going to happen? There will be a parent cell, and a parent cell will divide into two identical daughter cells here. In mitosis, a parent cell divides into two identical daughter cells here. Sir, what is the meaning of identical? Identical genome, of course. So a parent cell will be diploid here, and even the daughter cells will also be diploid here, guys. Perfect. And next thing here is that how many stages? What are the stages of mitosis? Remember, mitosis will occur in 4 stages, that is prophase, and then metaphase, and then anaphase, and then telophase. So prophase, metaphase, anaphase, and telophase. Lovely. Now, once you are done with mitosis here, let's have a revision of meiosis. See, of course, we are not going to learn about mitosis and meiosis in detail here, all the stages, how is the process taking place. That is not our topic here right now. I'm just trying to revise only those points which are required here. Is it okay? Now, let's go towards the meiosis. In meiosis, a parent cell will actually divide into two daughter cells. Okay, and in the very first stage, that is meiosis 1, what is going to happen? The diploid parent cell will divide into the haploid daughter cells. It means the number of chromosomes are reducing to half in meiosis 1. That is what I'm trying to put in your mind. This is extremely important. So number of chromosomes are reducing to half in which division? Is it meiosis 1 or meiosis 2? Remember, it is meiosis 1. Further, what is going to happen? This daughter cell will further divide into two more daughter cells here, and this daughter cell will be dividing further into two more daughter cells with haploid number of chromosomes, haploid here, as well as the haploid number of chromosomes here, guys. And this is meiosis 2. So first thing I want to put in your mind is that number of chromosomes will be reduced in meiosis, and that to meiosis 1. Okay. And now, my dear friends, what about the stages? They are the same again. In meiosis 1, it will occur in prophase 1, metaphase 1, anaphase 1, and telophase 1. Whereas in the meiosis 2, again it is prophase 2, metaphase 2, anaphase 2, and telophase 2, guys. It will occur in the same stages. Perfect. Now, this is what I want you all to keep in mind, and then we can comfortably and with confidence go towards learning about the gametogenesis. That is, first we are going to learn about spermatogenesis. So let's begin now with spermatogenesis first. First, I want you all to learn like two important points here. Number one, what is the location of spermatogenesis? Where exactly will spermatogenesis take place? Your answer has to be seminiferous tubule. Seminiferous tubule, sir. Then next, number two, what will be the duration of spermatogenesis? Like how much time is taken for spermatogenesis to be completed? It will be 72 to 74 days. And here I want my students to please concentrate on this one, sir. It is days. During our live classes, I have seen students making like silly mistakes. They'll be answering like 72 hours or 72 minutes or 72 weeks, something like that. Remember, it is days, 72 to 74 days. Perfect. Now, after learning these 2 points here, let's actually start with the process itself. Let's begin with spermatogenesis entire process. And I'm going to tell you in a very simple manner in the form of a flow chart. So let us cover each and every MCQ here, each and every point with the help of a flow chart here. First of all, Spermatogenesis will begin with what? It will begin with spermatogonium cells. Want you to be like more alert from here onwards. Why? Because terminology is like quite confusing here. Spermatogonium, spermatocyte, spermiogenesis, spermiation. So all this terminology looks like very much confusing. I want you to be like more alert from here onwards. Spermatogenesis begins with spermatogonium cells. Now, don't get confused here. Suddenly, out of the blue, from where are these spermatogonium cells coming from? To make you understand that, I have to actually go back and teach you the entire general embryology. Then you will understand that one. Why? Why? Because spermatogonium cells are coming from primordial germ cells, and then from the epiblast, and then from there to the yolk sac. From there, they'll be going to the genital ridge. If I tell you all these terms before itself, you'll go mad. You will not be able to understand like what is happening over here. That's the reason why I'll keep that part for the end. So for now, you just continue from here. Yes, spermatogenesis is beginning from spermatogonium. What are those primordial germ cells and all those things? We'll see at the end of general embryology. You will understand more better at that time. Now, these spermatogonium cells are first of all undergoing mitosis. But remember, it's like a repeated mitotic division taking place here. It's not like only one single mitosis. Almost it will be for 5 to 7 cycles of mitosis years. Spermatogonium will undergo mitosis for almost 5 to 7 cycles. Then it's gonna become primary spermatocyte. Primary spermatocyte. Now, what is the condition of primary spermatocyte? Is it like haploid or diploid? Of course, it will be still diploid in condition. Why? Because only mitosis has taken place till now. So, it is still diploid in condition. Now, this primary spermatocyte will start undergoing meiosis, and of course, it will undergo meiosis 1. And as the name indicates, the primary spermatocyte is going to become secondary spermatocyte. The primary spermatocyte is going to become secondary spermatocyte. And just now we have revised in our mind, meiosis 1 is the stage where the number of chromosomes are reduced to half. So, diploid primary spermatocyte becomes haploid secondary spermatocyte. And why I am stressing a lot here? Why? Because this has been tested in almost every exam and many a times, guys. It has been tested in almost every exam, whether it is NEET PG or AIMS exam. It has been tested many times. So, what is the stage in spermatogenesis at which number of chromosomes are reduced to half? So, it is primary spermatocyte to secondary spermatocyte. Primary spermatocyte to secondary spermatocyte. That is the stage where the number of chromosomes are reduced to half, sir. And in one of the exam, I remember there was a different terminology given, guys. Yes. At which stage there will be independent assortment of maternal and paternal chromosomes. Look at the terminology here. Independent assortment of maternal and paternal chromosomes. It's nothing but, yes, from diploid it is going to become haploid. Meiosis 1 only. And again it is at primary spermatocyte to secondary spermatocyte. If you wish, you can just make a note of this one. From primary to secondary spermatocyte, what is going to happen here, sir? There will be independent assortment. Independent assortment of maternal and paternal chromosomes. Independent assortment of maternal and paternal chromosomes. Perfect. Next one. Now it's only meiosis 1 that has been completed. So of course, the secondary spermatocyte will undergo meiosis 2. It is going to complete meiosis. So it will undergo meiosis 2 and it is going to form spermatid. Spermatids, yes. But I want you all to keep one thing in mind. The spermatids are still in the form of a normal cell. It has not taken the shape of a spermatozoa with that head, neck, middle piece and the tail. So these spermatids are still in the form of a normal cell, like a cell membrane with the nucleus in the center, with the cytoplasm and all. So now what is going to happen? The spermatid will undergo certain morphological change. So therefore, this spermatid will undergo morphological change to become spermatozoa with that head, neck, the middle piece and the tail. What is the name given for these morphological changes here? This is known as spermiogenesis. Be careful here, I'm not telling spermatogenesis, it is spermiogenesis. So I repeat for the last and final time, spermatid are still like a normal cell. They are undergoing spermiogenesis to form the actual spermatozoa with the head, neck and the tail. And that's all. We are done with the formation of spermatozoa here. And guys, no need of confusion here. If you see the entire process, if you see the entire flow chart here at once, then you can see here it's all about mitosis and then meiosis and then the morphological change here. That's all. So make it simple. So it's like mitosis once and then meiosis and then finally the morphological change. The spermatozoa is ready there. Now after this, what is going to happen? Let us try to understand the further story from here. Where exactly is this spermatogenesis taking place, guys? Whatever I have made you write down in the form of a flow chart, where exactly it is happening? In males, the male gonads will be actually testis. And inside the testis, it is further divided into lobules, many lobules. Inside those lobules, will be having the highly convoluted tubule, highly folded tubule known as seminiferous tubule. Let's take this seminiferous tubule out here. So the name itself is telling it's like a tube, seminiferous tubule. Inside this seminiferous tubule, you'll be having flattened cells which are known as germ cells. And in the middle of that, you'll be able to find the cone-shaped cells which are known as Sertoli cells. The cone-shaped cells which are known as the Sertoli cells. Now, we have been learning from our school days, Sertoli cells are the one which are actually helping in the process of spermatogenesis. So, today, let us try to find out how. Let me just zoom that one of the Sertoli cell here, sir. A cone shape or pyramidal shape Sertoli cell. Now, when you look at the Sertoli cell microscopically, in the membrane of the Sertoli cell, you'll be able to find certain pouches. And in those pouches, you'll be able to find all the developmental stages of spermatozoa. Like you'll be able to find the primary spermatocyte, secondary spermatocyte, the spermatid, and finally the spermatozoa will be present at the tip of the Sertoli cell here, sir. So if you're looking at the Sertoli cell microscopically there, in the membrane, you'll be able to appreciate all the developmental stages, guys. Okay. So finally at the tip, you'll be able to find the spermatozoa. Now, why am I telling you about all these things? You yourself can understand what should be the next thing happening here. What is the next thing going to happen here, guys? Yes, these spermatozoa are going to be released from the Sertoli cells into the lumen of the seminiferous tubule. I repeat again, the spermatozoa are going to be released from the tip of the Sertoli cell into the lumen of the seminiferous tubule now. And even that has got a name. It is known as spermiation. It is known as what, guys? Spermiation. Oh, it means that after the formation of the spermatozoa, now the spermatozoa will undergo the process of spermiation. Spermiation. By the way, what is the meaning of spermiation, sir? Spermiation means release of spermatozoa from Sertoli cells into lumen of seminiferous tubule. From Sertoli cell into the lumen of seminiferous tubule. Perfectly done. Now, once they are released, then what is the next thing that is going to happen? They will migrate further. Those spermatozoa are going to migrate from the seminiferous tubule. And finally, they will be going to epididymis. And believe me, my dear friends, epididymis is extremely important when it comes to your PGME exams. There are so many things happening over here. Let us put it in the form of one by one, all the points, guys. Question number 1. Where will storage of spermatozoa take place? Answer, epididymis. Storage of spermatozoa will take place in epididymis. Where will be the spermatozoa undergoing maturation? Again, the answer is epididymis. And then finally, where will be the spermatozoa gaining motility? It's going to become like more mobile, more motile. That is again epididymis, guys. So storage of spermatozoa will take place in epididymis. Maturation of the spermatozoa will take place in epididymis. And even it is going to become like more mobile, more motile in epididymis itself, guys. That's an extremely important thing. Now, let me tell you one important MCQ with respect to maturation of the epididymis here, sir. Maturation of the spermatozoa in epididymis. Let me tell you one important MCQ here. What is going to happen here? After the formation of spermatozoa in males, it will be actually stored in the epididymis. Finally, it is done. But there is something known as capacitation. Now, what is capacitation? Capacitation is actually the final maturation of spermatozoa. But the catch here, the point here is that it's not taking place in males. Remember, capacitation will take place in the female genitalia. It will be in the fallopian tube. So once the spermatozoa are deposited in the vagina, from the vagina, the spermatozoa will travel via the uterus and they will go to fallopian tube. In the fallopian tube, final conditioning of the spermatozoa will take place, and that is known as capacitation, guys. So please write down there first of all. One more point to include here is capacitation. Now, what is the meaning of capacitation here? It is final maturation of spermatozoa in fallopian tube. It is the final maturation of the spermatozoa in fallopian tube, guys, in fallopian tube. And to be more specific, it will be taking place in the isthmus part of the fallopian tube. That is the final maturation. Now, what I am trying to tell you here, what is that important MCQ where most of the students are getting confused? There is a question given, maturation of spermatozoa takes place in dash. Two options are given, both the options are given, epididymis, and option number 2 there is actually fallopian tube. Now the students are confused which one to be marked, where is maturation taking place. Remember, whenever this type, if you come across this type of question, the best answer will be always epididymis. Why? Because actual maturation of the spermatozoa is taking place in epididymis only. In fallopian tube, it is just capacitation, and that is just the conditioning of the spermatozoa, the final maturation of the spermatozoa. Is it okay? So if you have to select an option between these two, the best option will be epididymis. Actual maturation of the spermatozoa is taking place in epididymis, guys. Is it okay? So that completes your entire spermatogenesis over here. Now, one last and final question which is remaining here to be answered is, whenever you are going to begin with one spermatogonium, if you are beginning with like one spermatogonium, at the end, how many spermatozoa will be formed? How many spermatozoa will be formed from one spermatogonium? So remember, it will be 64 to 512 spermatozoa formed from one spermatogonium cell. So why does this variable number here? Why? Because it depends on this mitotic cycles here. If it is 5 cycles of mitosis taking place, then it will be 64 spermatozoa formed here. And if it is 7 cycles of mitosis taking place over there, then it is 512 spermatozoa formed there. So that is about the number. And by this, we complete the spermatogenesis. Each and every point has been covered here in a crystal clear manner, guys. According to our planning, let's move on to oogenesis right now. So spermatogenesis is done. Let's move on to oogenesis. Now, when you're learning about oogenesis, guys, you got to be more careful. Why? Because in females, oogenesis is going to begin during intrauterine life. In males, the spermatogenesis will begin at the time of puberty only, after birth, puberty. But in females, the formation of the ova will begin from the intrauterine life itself, before the birth. So we need to start from there, right from the intrauterine life, what's happening. Then we need to learn before the birth what's happening. Then we need to learn after the birth, during childhood what's happening, and then up to the age of puberty. So we have to see the entire timeline right from before the birth up to the puberty. Okay, that is why you got to be like more careful here in oogenesis. But there is even a good news here. The good part here is that it's almost similar. How come? We started spermatogenesis with spermatogonium, oogenesis, oogonium. And then spermatogonium will get converted to primary spermatocyte, primary oocyte, secondary spermatocyte, secondary oocyte. So in that manner, it is a little bit easier, guys. Okay. So without any further delay, let's get started right now. Oogenesis is going to begin with oogonium cells. And these oogonium cells are undergoing mitosis. Okay. And once they undergo mitosis, we know very well they are going to become what? Primary oocyte. Primary oocyte. Now again the same question here. What is the condition of the primary oocyte right now? The condition of the primary oocyte is it's still diploid. 2n number of chromosome. It is still diploid. because only mitosis has taken place till now. Reduction division, that is meiosis, has not yet taken place. Now the primary oocyte will begin the process of meiosis, and that too meiosis 1. Primary oocyte will start undergoing meiosis 1. But the important question here, will it complete meiosis 1? No, it's not going to complete the meiosis 1 here. It will be arrested. That is what is something important to remember here, guys. It will be arrested in which phase of the meiosis 1? Remember, primary oocyte will be actually arrested here in prophase 1. Which stage, guys? Prophase 1. And if you are further interested in this one, to learn in more depth, in prophase 1 also, remember, it will be arrested in diplotene stage. Diplotene stage. So let us learn like step by step. So primary oocyte will be arrested in prophase 1, and that too diplotene stage of the prophase 1, guys. I hope now you will be completely agreeing with me. In the very beginning of this topic, why did I recall you about the mitosis and meiosis? See how confidently we can learn here. Now, it's arrested here. It is no more further development taking place. Now we need to understand the timeline here. This is the place where most of the students will be getting confused here. We need to understand the timeline. When is all this happening? All this is happening like before birth. In the intrauterine life, before birth. Okay. So it is arrested in the primary oocyte form only before the birth. Now what is going to happen further? Further development of the fetus will be taking place. It will be still in the primary oocyte form only. Then finally, the day will come when birth of the female will take place. So at the time of birth also, it will be in the form of primary oocyte. Then the age of that baby will keep on increasing. During childhood, again it will be in the form of primary oocyte, arrested in prophase 1. Then finally, at the time of puberty, what is going to happen, guys? It's going to complete prophase 1, then metaphase 1, anaphase 1 and telophase 1. I mean to say in simple words, it's going to complete meiosis 1. Understanding? So let us just register all those stages one by one. So that's happening before the birth, and then even at the time of birth also, what is going to happen, guys? It will be in the form of primary oocyte only. After birth, even during the childhood also, it will be in the same form, the primary oocyte only. And the next important landmark right now is puberty. Now at the time of puberty, what is going to happen here? It completes meiosis 1. Completes meiosis 1. And once the meiosis 1 is completed, it will lead to the formation of secondary oocyte. It will lead to the formation of secondary oocyte, and obviously the secondary oocyte will be haploid. You can just look at that entire time period, guys, starting from before the birth, entire development during the pregnancy, birth, and during the entire childhood, and somewhere at the age of puberty, what is going to happen? The primary oocyte will get converted to secondary oocyte. Have a clarity in that. And along with the secondary oocyte, it is going to form the first polar body also. The first polar body will also be released along with the secondary oocyte, guys. Next, further, what is going to happen? Meiosis 1 is completed. Now it has to complete meiosis 2. So therefore, this secondary oocyte will now start undergoing meiosis 2. But again, the same question here. Will it complete meiosis 2? Absolutely no. It will be again arrested. And you got to remember, this time it is arrested in which phase? It will be arrested in metaphase. And of course, it will be arrested in metaphase 2. So keep it very clear in your mind. Before the birth, during intrauterine life, it is arrested in prophase 1. Whereas after the birth, it will be arrested in metaphase 2. Metaphase 2. Automatically, the next question coming in our mind, then what is the fate of this one, sir? When it is going to complete that meiosis 2? What is the fate of this one? The fate of the secondary spermatocyte will actually depend on two things, sir. Number one, the availability of spermatozoa, or if it is not available, then what is going to happen? So let us take condition number 1. For example, if the spermatozoa is available, sir, I'm just writing plus here. If the spermatozoa is available, then what is going to happen? It is going to complete the meiosis 2. Fertilization will occur. It is going to complete the meiosis 2. And it is going to become like ovum along with the release of the second polar body, and further pregnancy will actually continue, guys. So let us write down. So if the spermatozoa is available there, or we can say it completes meiosis 2, if fertilization occur. Fine. So it completes meiosis 2 only if fertilization will occur. So when it completes meiosis 2, what else is going to be formed there, sir? At the time of fertilization, it will form the ovum along with the second polar body. So if the spermatozoa is available, of course fertilization will occur, and at the time of fertilization, it is going to complete meiosis 2 with the release of ovum along with the second polar body, guys. That's one case. Welcome to the case number 2. Suppose if the spermatozoa is not available. If the spermatozoa is not available, then what is going to happen? Yes, it's going to persist as corpus luteum. So corpus luteum will actually persist, and then it is going to persist for another 14 days of the menstrual cycle. And at that time, what is going to happen? It is going to become like a scar, which is known as corpus albicans. That's all. Again, the next second menstrual cycle is going to begin. No pregnancy, nothing. Understanding? So if there is no spermatozoa is available, it persists as corpus luteum. Corpus means the body, luteum the yellow colored one. It persists as corpus luteum, and then it is going to persist for another 14 days. Okay, and after that 14 days, there will be just a white scar there known as the corpus albicans. That's the fate of the secondary oocyte. It depends if fertilization occur, and if fertilization not occur, then what is going to happen, guys? So I just want you to remember one more important thing here, that when is the meiosis 2 going to be getting completed? It will be getting completed only if fertilization will occur. Otherwise, it's going to continue the menstrual cycle. And the last and final thing that I want you to stress here, one more important thing that I want to stress in your mind is that please don't forget the stages at which it is arrested. Remember, before the birth, during intrauterine life, it is arrested in prophase 1. And after the birth, it will be actually arrested in metaphase 2 of meiosis 2. That's something really important, will be tested many a times in many exams. So that completes the spermatogenesis and oogenesis. So basically we are done with the gametogenesis. And according to our planning, now we can move ahead in general embryology with the first week and second week and third week of development, guys. So let's begin with the first week of development now. Now let's continue with the first week of development, guys. And I want, first of all, my students to have a clarity, like what's happening in the first week. During the live classes, when I ask the students, like what's happening in the first week of pregnancy, random answers will be given like zygote, morula, these, that and all. First, have a clarity, okay, what's happening in the first week of pregnancy, guys. There are two important events taking place. Number one, fertilization, and number two, implantation. So what is going to happen in the first week of pregnancy, guys? That is fertilization and implantation. These are the two important things happening here. And I'll first explain you with the help of a diagram here, and afterwards we'll be writing the notes in the form of a flow chart. Okay, let's draw a beautiful diagram for this one. So imagine this one to be the fallopian tube here, and this is the uterus here, sir. The fundus, body, and the cervix part of the uterus, followed by the vagina. So imagine that is like, I'm just drawing half of the diagram there. One fallopian tube along with the uterus and the vagina part. Now, for example, normal menstrual cycle, a regular normal menstrual cycle will occur for 28 days. And exactly on the 14th day of a regular menstrual cycle, ovulation will occur. So, for example, you just imagine that time right now. So, we are at the 14th day of the menstrual cycle and there will be ovulation taking place. And the ova will be expelled out, and that ovum will be taken up by the fallopian tube here. So imagine this is like ovum, and ovum is actually surrounded by a layer that is known as zona pellucida. So whatever I have drawn there with the green color there, that is zona pellucida. So on the 14th day of the menstrual cycle, a regular menstrual cycle, there will be ovulation occurring, and ovulation, ova will be expelled out, taken up by the fallopian tube right now. Suppose at the same time if spermatozoa is available there, then what will happen guys? Fertilization will take place. And once the fertilization will take place, what is the result of fertilization? What is going to form here? Yes, there will be formation of a zygote with that ovum, the nucleus of the ovum, the nucleus of the spermatozoa. So there is a formation of a zygote, and this zygote is still surrounded by zona pellucida. Now, is there any MCQ which is possible here? Yes. You need to remember when will fertilization take place, on which day. And remember, my dear friends, fertilization will be taking place on day 1. So I'm just going to represent the days with D there, day 1. Fertilization is going to take place on day 1. Or vice versa, opposite of that one, if someone asks you, like which should be considered to be the first day of the pregnancy? The day on which fertilization occurs. So the day on which fertilization occurs should be considered to be the first day of the pregnancy. Now further development. Zygote will further multiply, and it is going to multiply to form almost like 16 cells right now, sir. And when there are totally like 16 cells, it will appear like a mulberry. That embryo will actually appear like a mulberry. So that is the reason why it is said to be morula. So zygote will multiply to form morula. Now, is there any MCQ possible here? Of course. How many cells do you have in the morula? Remember, morula actually has got 16 cells. But there is a difference of opinion, guys. In variety of books, when you refer to different embryology books, it's given like 8 to 16, 16 to 32, somewhere 8 to 32. But one common number is 16. So 16 cells will be there in the morula. Now this morula will further multiply, and those cells will further multiply to form almost like 32 cells in that one cell. And it's surrounded by zona pellucida. What is the name of this stage here? This is known as the late morula, or else it is also known as the late or advanced morula. Late or advanced morula. Now there are two important MCQs which are possible here, guys. Number one, what is the stage in which the embryo enters from the fallopian tube into the uterus? Is it like zygote entering, or the morula, or blastocyst? Like what is entering from the fallopian tube into the uterus? Remember, the stage which will enter into the uterus will be the late morula or the advanced morula. So late morula or the advanced morula is the one which is going to enter from the fallopian tube into the uterus. Question number 2. On which day? Remember, it is going to enter into the uterus on day 4, 4th day. Now, once this late morula enters into the uterus on 4th day, then further what is the development, guys? These cells are going to rearrange themselves into a layer of cells on the outer side, and that is referred to as outer cell mass. So, this will be actually the outer cell mass. I'll just write down in short here as OCM, outer cell mass. And there'll be some more cells present over on the inner side here, sir. And these cells are actually referred to as your inner cell mass. So there will be a layer of cells on the outer side, that is outer cell mass, and there'll be some more cells present inside, that is inner cell mass. And this is still surrounded by zona pellucida. Zona pellucida. Now let's have a discussion. Let's speak regarding zona pellucida, guys. From that time, you might be even observing. Okay, I'm going on drawing the zona pellucida in the zygote, morula, advanced morula. Zona pellucida is present over there. But what is the reason? Why? What is the function of that one? Remember, zygote and morula and all, they are very much sticky. The moment they will come in contact with any surface, they'll stick. So, for example, if this morula sticks to the fallopian tube, surface of the fallopian tube, then what will happen? Implantation will occur there. Is it normal? No. It is actually ectopic pregnancy. It will lead to ectopic pregnancy, and that has to be avoided. And that responsibility will be actually taken care by zona pellucida. So that zona pellucida has got the responsibility of taking that embryo safely into the uterus and then get it implanted over there. So we can say it is the one which is preventing ectopic pregnancy. Number one function. Number two, not only ectopic pregnancy, it is also preventing polyspermy, guys. Like one spermatozoa fertilized, further there should be no other spermatozoa fertilizing that one, and that is also prevented by zona pellucida. So number one, zona pellucida prevents polyspermy, and number two, remember, it also prevents ectopic pregnancy. Ectopic pregnancy, guys. Now, again, try to recall, like, what are the two things which are taking place in the first week of the pregnancy, guys? Fertilization and implantation. Now, out of these two, I am already done with the fertilization. Now, what about implantation? Now, we got to learn about implantation. I think the way I told you here in a conceptual manner, you yourself will be able to apply the concept right now. Oh, who is preventing the implantation here? Zona pellucida. So what is the next thing that should happen here right now? That zona pellucida has to be gone. It has to be degenerated for implantation to occur. So now this zona pellucida layer will be degenerated, which is actually referred to as zonal hatching. Okay. Outer cell mass will be present there itself. No problem with that one. Okay. The outer cell mass layer will be there only, guys. And now what will happen, because the zona pellucida is gone, now the fluid will get the chance to enter inside. Which fluid am I speaking about? Obviously, we are in the uterus here right now. So uterine fluid will enter inside. And the moment the uterine fluid will enter inside, the inner cell mass will be pushed aside. So this inner cell mass will be actually pushed aside right now, guys. The moment the uterine fluid will enter inside, the inner cell mass will be pushed aside. And now you can clearly observe there, it's appearing like a fluid filled cavity. And in our entire MBBS, the fluid filled cavity is referred to as cyst. And that's the reason why it is known as blastocyst. This is blastocyst, fluid filled cavity. Learn with the meaning, guys. Okay, don't try to just memorize the names simply just like that. Why it is morilla, why it is blastocyst? If you learn with the meaning, then you will be able to retain that for a longer time. So this is appearing like a fluid filled cavity there, blastocyst, and obviously blastocyst doesn't have the zona pellucida there. So therefore, this is the stage which can be implanted into the wall of the uterus, guys, in the endometrium. It will be implanted. Perfect. So we are done with the fertilization. We are done with the implantation here. That completes your first week here. First week of the pregnancy has been completed in a very crystal clear manner in this diagram here. And almost every MCQ covered here. The moment you look at this diagram, you'll be easily able to revise the entire topic, guys. Okay. So just to complete the formality here, let us just do one thing. Write it in the form of a flow chart right now. So try to recall what's happening on the day one. On the day one, fertilization. What is the result of fertilization? It will lead to the formation of zygote. Zygote will further multiply to form what's a morilla. Morilla will have how many cells? 16 cells. This morilla will further multiply to form the late or advanced morilla. It is going to form late or advanced morilla, and that will consist of 32 cells. Something important here, guys. Late morilla is the one which will enter uterus. Which stage is going to enter? Late morilla will enter the uterus. On which day? That is day 4. Now once it enters, finally it is going to form what's a blastocyst. With all those changes taking place, it is going to become blastocyst. Perfect. Now this blastocyst is also very much important for your exams here. Many questions, many times the questions will be asked about the blastocyst. So let us just write down this one here. Question number 1. What is the stage of implantation? The stage of implantation will be blastocyst. What is the stage of implantation here, sir? That is nothing but blastocyst. Question number 2. Implantation begins on which day? Implantation begins on which day? Remember, implantation begins on day 6th. 6th day. Again, the same thing. They are like variable numbers given in the books, guys. The correct answer, the final answer will be 6th day. Number 3. Implantation completes by which day? Implantation completes by 10th day, day 10. And then next, one more thing. These are all things are done. Now, what is the site of implantation? Where exactly implantation is taking place here? Site of implantation. Remember guys, implantation will be taking place at the junction of fundus and body of the uterus. Fundus of the uterus and the body of the uterus at the junction of that one. But is it on anterior wall or the posterior wall? Remember, it's on the posterior wall. So, where exactly the implantation will take place? It is at the junction of fundus and body of uterus. At the junction of fundus and body of the uterus, on which wall? On posterior wall. On the posterior wall of the uterus. So, implantation will occur at the junction of fundus and body of the uterus on which wall? On the posterior wall of the uterus. So, these are all the points that you have to remember about the blastocyst or in relation to that one, guys. So, what is the stage of implantation? It's blastocyst. Implantation is going to begin on which day? 6th day is the right answer. And finally, it is going to complete by which day? 10th day. And what is the site of implantation? Junction of body and the fundus on the posterior wall, guys. Posterior wall of the uterus. So, that is about the first week of pregnancy, guys. And from here onwards, we have to move towards the second week of pregnancy. And we got to continue what is the further changes that are taking place in blastocyst, that too in the outer cell mass as well as inner cell mass. And that we are going to further see in the second week.